This is gonna be week 3, like a big summary. So essentially, this is our testes right here, and your seminiferous tubules are what are making essentially the sperm. And what they're doing is they're having something called asynchronous production. This is because the sperm life cycle, or in order to get the mature sperm, is around 72 days. So if they were not asynchronously producing sperm, or if there were all producing sperm at the same time, what would end up happening is that a male would be fertile every 72 days. Now, this poses a problem because let's say they're able to find a mate on one day, but then they don't have enough sperm to fertilize the mate. This would be an evolutionary disadvantage. So if you look at the seminiferous tubule, what you'll see different sperm cells at different stages. And what this allows males to do is that despite the life cycle or the amount of days that it takes to get to maturation being 72 days, they're able to fertilize a mate every other day. So essentially, these are your seminiferous tubules. They feed into the rete testis, which then goes to the caput, which is the head of the epididymis, the cauda, which is basically the base of the epididymis, and then the corpus. Now, the cauda's purpose is that it essentially serves as a pouch where sperm is stored, right? It goes up through the corpus, caput, and the epididymis as a whole is where sperm are going to undergo their maturation. Now it's going to come all the way down, and we're going to hit a couple glands, if you will. So we have the seminal vessels. These are going to secrete fructose, prostaglandins. Prostaglandins usually help with vasodilation. This is around 70% of the ejaculate volume. Prostate, 30% activates sperm and also excretes citrates. And then you have your bulbourethral glands, which secrete this thick, clear, alkaline mucus. The reason for this is urine is actually toxic to sperm when acidic, so it helps neutralize the urine. Then looking at your seminiferous tubules more, remember this is where the sperm is being produced. We have a couple cells and histology that we do need to know. And you will need to be able to recognize these under a slide, so I think that's part of the GLS. Leydig cells. These are the cells that are going to be producing the androgens, which are the testosterone in this case. We have myeloid cells, so they're contracting to move sperm. And we have our Sertoli cells, which are just promoting and supporting spermatogenesis. Okay? So Leydig cells, they're producing the androgens, the testosterone. Myeloid cells, they're contracting to move the sperm. Sertoli cells, they're supporting spermatogenesis, S and S. So now let's talk about the main or bigger hormonal control. GnRH is stimulating the anterior pituitary. It's then releasing FSH and LH. LH to your interstitial cells, and FSH to your Sertoli cells. Remember, what are Sertoli cells doing? Again, what are Sertoli cells doing? They're nourishing sperm precursors. They're releasing spermatid into the lumen, and they phagocytose excess cytoplasm, right? So those are the functions of your Sertoli cells. Now, FSH is also going to trigger ABP and inhibin, and this is just a negative inhibitory pathway on the anterior pituitary to decrease FSH and LH. Now, the testes in the male anatomy are basically in this pouch, and this pouch has smooth muscle that can essentially contract or relax to bring the testes closer or further away from the body, and this serves to essentially control temperature. Now, the reason for this is that sperm are happy around 4 degrees less than body temperature. So if body temperature is 37, let's say 33. And there's another component that we do need to learn, which is the paniform plexus. This wraps around the testicular artery, and due to the artery having higher blood flow than the plexus or the veins, this can help reduce blood temperature even more. Now let's talk about spermatogenesis, and essentially its precursor cell production. So you're going to start off with your type A daughter cells. This is again going to turn to type B daughter cells. What's the purpose of the type B daughter cells? Well, you don't want to get rid of your type A supply, so you make another type of precursor cell. So that, therefore, you have your type B daughter cells. Then you have your primary spermatocytes. They move out from the basement membrane into the adluminal membrane. They turn to secondary spermatocytes. Then you have your early spermatids, late spermatids. This is your system. I honestly would just watch a YouTube video on 2N and 4C. It's way easier to explain that way. But basically 2N4C for your type A daughter cells, 2N2C for type B, 2N4C again because you're just duplicating the cells again. You're then going to 1N2C because this is meiosis 1. And then because this is meiosis 2, you're going from 1N2C to 1N1C. This has to do with like chromosomes and chromatids. And honestly, a YouTube video would just explain it better than me trying to attempt an explanation. Spermiogenesis is when you're going from the early spermatids to late spermatids. And essentially what's happening in spermiogenesis here from the early spermatids to late spermatids is that you're forming this spermatid cap. And I wanted to go into more detail about that. So the first phase is a cap phase. And DNA is going to be tightly packed. And the Golgi apparatus is going to form around the vesicle. So it's going to create this mushroom head, essentially. The second phase is the acrosomal phase. And this is where the nucleus and cytoplasm is going to elongate. And you're going to get this type of shape right here. And then the maturation phase is where mitochondria are going to form in the midpiece. And the tail is going to mature to have these nice flagella that allow this sperm to essentially swim. Now, these three things are part of spermiogenesis, which is from early spermatids to late spermatids. Meiosis 2 is from secondary spermatocytes to early spermatids. Meiosis 1 is 2N4C to 1N2C, from the primary spermatocyte to the secondary spermatocyte. The primary spermatocytes are coming, they're essentially going from the adluminal compartment. They're coming from the basement membrane. Why do we have type B daughter cells? This is because we don't get rid of our stem cell supply, if you want to call it that. And that is the reason for that. So your type A daughter cells are splitting into two cells, hence the 2N4C to 2N2C. These are hormonal controls. Asynchronous production, 72 days. Why? This is because you want to be fertile throughout the 72 days, not just once every 72 days. Leydig cells produce androgens. Sertoli cells support spermatogenesis. Myeloid cells contract and move sperm. And GnRH is going to release, is going to go to the anterior pituitary. It's going to release FSH and LH. LH to your interstitial cells, FSH to your Sertoli cells. FSH is also going to stimulate ABP and inhibin. Inhibin, like the name suggests, is going to inhibit the anterior pituitary as a negative feedback mechanism. You have your seminal vessels, prostate, bulbourethral glands, in that order of, I guess, the spermatocytes moving. This is the anatomical order, essentially. So you're going from 70-30, not really adding anything here except alkaline mucus to neutralize the urine. Prostate, citrus, activating sperms, seminal vessels, fructose, prostaglandins. That serves as a vasodilator, 70% ejaculate volume.